Good afternoon and welcome to this seminar, which is in the frame of the newly appointed faculty member seminars organized by Humanitas University to welcome the new members of the faculty and to present them to the students, which unfortunately today are not so many, but you know they are quite busy studying for the exam in this period. And this is the reason why we will stop this seminar until the beginning of October, where we will have a long queue of colleagues that are eager to present their seminar. Because, as you may know, recruitment is going on at very high speed in Humanitas University in these months. Today I have the privilege to present you a young scientist who has been working here for the past 9 years or something, so he's well known, but he's assistant professor in general pathology from January 2016 in Humanitas University. Let me briefly summarize his CV and then I will leave him the stage to present his seminar. He got a degree in cellular biology at the University of Poitiers in France in 2004, and then he got a PhD in immunology, moving to the University of Angers at INSERM. He did 1 year of postdoc in the same institution, and then he moved to Humanitas as a postdoc and then as a junior group leader in the Laboratory of Immunology and Inflammation. He received a postdoctoral investigation award from the International Cytokine Society in 2012. And his research activities has mainly focused on the molecular and cellular mechanism of innate immunity and inflammation in health and disease. He is young, but I went through his biosketch and I saw a number of significant achievements, which I will just briefly summarize. He gave important contribution in understanding the immunoregulatory properties of neutrophils, in particular in the activation and regulation of innate and adaptive immunity. And he worked a lot on the long pentoxin-free, PTX3, in particularly on its antimicrobial properties, in its role in mediating the clearance of apoptotic cells, in its role in the resistance against urinary tract infection, demonstrating that PTX3 polymorphisms are associated with susceptibility to acute pyelonephritis, on the role of PTX3 deficiency in neonates, in the susceptibility to infection diseases, and in the importance of milk-associated PTX3 in protecting from infection, the neonates, and probably the most important achievement, which was the demonstration that PTX3 acts as an oncosuppressor, inhibiting the complement-mediated tumor promotion. So a number of significant achievements, and he also has additional studies in the identification of PTX4 and in the study of infiltrating cell in colorectal cancer. So many scientific interests that I think he will summarize today in his seminar. So thank you, and I leave you the stage.
Thank you, Stefano, for this introduction. So good afternoon, everybody. Today, as mentioned by Stefano, we'll talk about neutrophils, and in particular neutrophils in immunity and inflammation. And we will talk in the role of neutrophils in inflammation and in cancer. But to talk about neutrophils, we have to begin at the beginning. And the beginning of neutrophils is the birth of immunology. And the birth of immunology at the end of the 19th century was accompanied by a great controversy between two theories. Theory of cellular immunity, mainly led by Elie Metchnikoff, and theory of humoral immunity, mainly led by Paul Ehrlich and German school. Briefly, Eli Metchnikov, we know now that Eli Metchnikov described the phagocytosis, and he claimed that immunity and defense against pathogen, the base of the defense of pathogen was cellular immunity, and the base of defense against pathogen was phagocytosis. And this is a cartoon made by Eli Metchnikov. Here it's macrophages, so we know the cells, with still the same name today, macrophage. And here it's microphage. And microphage, it's neutrophil. Because to have a macrophage, you need macrophage. And macrophage was a neutrophil. So it's interesting to note that Paul Ehrlich was one of the first scientists to describe immune cells, even if he defends humoral immunity. And a student of Eli Metchnikov was the first to obtain immune serum against endotoxin. So now we know that neutrophils are not just a phagocyte and not just a cells that patrol blood, looking for microbes and infiltrate tissue and infiltrate inflamed tissue. We know that neutrophils sustain immunity. We know that, of course, it's true that neutrophil is a phagocyte. And we know now that neutrophils express a lot of cytokine, chemokine, able to regulate innate and adaptive immunity. And there is a lot of crosstalk between neutrophils and other leukocytes. In particular, we also know that neutrophils express a set of pattern recognition molecules. These molecules are specialized in recognition of motifs expressed by pathogen, and these motifs were called PAMs, for pathogen-associated molecular patterns. The most famous example of PAMs is LPS, and based on their localization, these molecules were divided between cell-associated molecules, where we found capturing receptor, capturing molecules such as scavenger receptor or Fc gamma receptor, sitaplektin, and signaling molecules, where we can find the toll-like receptor. At the opposite, we have here the humoral pattern recognition molecule, which form with the complement component, the humoral arm of innate immunity. And inside this superfamily of molecules, you have three subfamily, phycolin, colectin, and pentraxin. And today we'll talk about pentraxin, and in particular about one member of this family, which is PTIC3. So pentraxin is a superfamily of evolutionary conserved molecule, and based on their primary structure, pentraxin were divided between short pentraxin, so SAP and CRP, which constitute the acute phase molecule in mouse and human respectively, and the long pentraxin, where PTIC3 was the first member clone identified in the early 90s by the group of Alberto Mantovani. And this is the structure of PTIC3. So PTIC3 is a secreted molecule with a classical signal peptide, and is coupled with a C-terminal pentraxin-like domain and an N-terminal unrelated N-terminal domain. This is the structure of PTIC3, which is mainly found as covalently linked octamer, supported by a network of disulfide bond. What is the biological function of PTIC3? We know that PTIC3 can exert function in numerous environments. So first, PTIC3 is expressed by numerous cell types, such as epithelial cells, endothelial cells, leukocytes, dendritic cells, macrophages. We will talk also about neutrophils, of course. And this is induced after stimulation by pro-inflammatory cytokine, so TNF alpha, interleukin-1 beta, mostly. Also toll-like receptor agonist, also pathogen. And PTIC3 is involved in matrix deposition, female fertility. Last year it was published in GXMed paper by Andrea Doni also that PTIC3 play a role in tissue repair and wound healing. PTIC3 is involved also in angiogenesis, mainly to inhibition and binding to FGF2 in complement activation. And last year, Eduardo Bonavita in our group published in a Cell paper showing that PTIC3 act as an oncosuppressor gene that can regulate complement-dependent inflammation in cancer. PTX3 can also block recruitment of neutrophils through binding to P-selectin. And this is what I will talk about today. PTX3 is highly involved in innate immunity, in recognition and elimination of selected pathogen. So some years ago, we published that PTX3 was constitutively expressed in neutrophils. And neutrophils release PTX3. This is a constitutive expression by FACS and microscopy. And PTX3 is very rapidly released after stimulation by toll-like receptor agonists, pro-inflammatory cytokine, or also directly by microbes. And this constitutive expression is consistent with the fact that PTX3 is stored in secondary granules. So granules which are many found as gelatinous low lactoferrin high granules. Some years ago, the group of Arturo Zingliski and Volker Brickman demonstrated the formation of an extracellular fibrillary network by neutrophils, and this network was called NETs, for neutrophil extracellular traps. These nets are formed by DNA, but in general by nuclear component, and are decorated by protein found in neutrophil granules. For instance, it was described that PTX3, not we described that PTX3, but it was described that protein 3 and MAPO was found in nets, and we also found that PTX3 was found in the structure. It's important to note that nets, the main activity of nets was to trap bacteria, to trap pathogen, and to kill them inside the nets. So we know that PTX3, and it has been published by the group of Alberto Mantovani, that PTX3 deficient mice present a high susceptibility to Aspergillus fumigatus infection. And interestingly, PTX3 had therapeutic activity because injection of PTX3 can rescue the phenotype. So we ask whether neutrophil-derived PTX3 has a role in this protection. And in order to demonstrate this, cyclophosphamide wild-type or knockout mice were infected by Aspergillus fumigatus and received 1 million of neutrophils, wild-type neutrophils or knockout neutrophils, also macrophages and T cells in control. And then we, 2 days after, we quantified the fungal growth. And as you can see, only injection adoptive transfer of wild-type neutrophil decreased the fungal growth. So this clearly demonstrated that neutrophil-derived PTX3 play a fundamental role and is essential for elimination of this pathogen. What's about effector mechanism in this model? We know that PTX3 can bind to conidia from Aspergillus fumigatus. And this binding of this opsonization increases the recognition and phagocytosis of conidia by neutrophils. This occurs in a serum-dependent manner, but immunoglobulin-independent manner. And to make a very long story short, what it has been shown by Federica Moali in our group is that PTX3 can bind to Fc gamma receptor 2, and when PTX3 binds to Fc gamma receptor 2, you get an activation, an inside-out activation of the CR3, complement receptor 3, CD11B, CD18. And this is a receptor for C3b. And so, by this activation, you enhance the phagocytosis of C3b opsonized conidia. So what you get, it's an amplification loop of the elimination, the recognition and elimination of the pathogen. Then subsequently, it was also been found that PTX3 has also therapeutic activity against Pseudomonas aeruginosa. And this is probably due to the same mechanism because it was reported here in GI that therapeutic activity of PTX3 was lost in FI3 knockout mice, but was conserved in C1q knockout mice. It was also lost in Fc gamma receptor knockout animals. We also demonstrated that PTX3 has therapeutic activity in neonate mice infected by Pseudomonas aeruginosa. Here, we don't investigate the molecular mechanism. So, in a more general view, what this story showed is the role that neutrophils can play in humoral arm of innate immunity. Because neutrophil can store this molecule. It stored PTX3, but can also store some member of ficolin family. And this allows the release, the expression of this molecule very, very, very rapidly after infection, in minutes. And this precedes expression of this molecule by other cell, where the expression is dependent of the gene expression. Other cells are other leukocytes, macrophages, dendritic cells, but also epithelial and endothelial cells. Now I will speak about another model of infection, more recent model. This is model of urinary tract infection. We know that urinary tract should be sterile and is normally sterile, and despite increasing usage of antibiotic in population, urinary tract infection remains one of the most common infections in humans and causes considerable morbidity such as renal failure, sepsis, until death in particular population. Pathogens which induce this infection are mostly E. coli and called UPEC for uropathogenic Escherichia coli. And this pathogen can bind to epithelial in the bladder to cause cystitis and can ascend to the kidney to cause pyelonephritis. We know that the host responds rapidly to this infection. The first response is exfoliation of cells, and here probably neutrophils are involved by production of reactive oxygen species that induce detachment of epithelial cells. This induces elimination of a high number of infected cells, but this also induces possible contamination of the most immature underlying cells. You also have expression of cytokines, chemokines, which support recruitment of leukocytes, and in particular recruitment of neutrophils. And you have expression of antimicrobial peptides, such as cathelicidin. But it was really strange for us, that the role of the humoral arm of innate immunity in this kind of infection was totally unknown. So we set up a model of urinary tract infection in wild type and PTX3 knockout mice by transurethral infection. And this is what we follow the bacterial burden in mice at different time points, at day 1 and day 5 in the bladder and the kidney. And this is what we observe. At day 1 post-infection, we didn't see difference in terms of bacterial burden in bladder. And this is consistent with the fact that PTX3 is not involved in the first attachment of bacteria to epithelium. In contrast, at day 5 post-infection, whereas the bacterial burden was decreased in knockout mice, it remained very high in knockout animals, at the same level that the one found at day 1. So this showed that PTX3 deficient mice presented the effect to control the infection. Also here, what's about some mechanism of protection, and also here we found that PTX3 was able to bind to UPEC, and binding in a dose-dependent manner. And this binding increased phagocytosis of UPEC by neutrophil. What is the source of PTX3 in the urinary tract infection? This is immunohistochemistry analysis of PTX3. In a basal condition, we didn't see expression of PTX3 both in bladder and the kidney of very, very, very low level. And in contrast, at very early time point after infection, so 6 hours after infection, we found very high expression of PTX3 in epithelial kidney cells and also in inflammatory cells infiltrating the bladder. And at 24 hours after infection, we found expression of epithelial cells both in the kidney and the bladder. So this is also consistent with the fact that we found that non-malignant urothelial cell line, human cell line, produce PTX3 at very low level, at basal level, but this expression increased very, very rapidly and strongly after stimulation by pro-inflammatory cytokine, by toll-like receptor agonist, but also directly by UPEC strain. So next, we determined the signaling pathway involved in this induction, and MyD88, THRIF, and toll-like receptor were silenced in this cell line, and as you can see here, only silencing of MyD88 and TLR4 here abrogated the production of PTK3. And this is also consistent with the fact that if you infect MyD88 knockout mice with UPEC, we didn't see increased expression of PTK3 mRNA. In contrast, if you infect THRIF knockout mice, we found increased expression of PTK3 mRNA. So this demonstrates that UPEC induced PTK3 in epithelial cells through a TLR4 mediated pathway. What about inflammatory response? We have a look about in inflammatory response because we know that balance between activation of innate immunity and inflammation and regulation of inflammation is a key for a good response in urinary tract infection, because you need inflammation, you need activation of innate immunity, but you need that this activation should be controlled. And this is the myeloperoxidase found in kidney and bladder, and neutrophil found in urine. So myeloperoxidase quantified the infiltration of leukocyte in tissue. And as you can see here, wild-type mice are in white. We found high and early recruitment of neutrophils of myeloid cells at early time points, both in wild-type and knockout animals. But this inflammation is transient in wild-type mice, and inflammation was very, very minimal in wild-type mice at D5. In contrast, inflammation remains very high in knockout mice. So this shows that increased susceptibility is associated with an exacerbated and uncontrolled inflammatory response. Of course, that can lead to tissue damage. What's about human here? We know that, and some group published, that genetic polymorphism were found on PTK3 gene, and this polymorphism were associated with clinical parameters, in particular susceptibility to Pseudomonas aeruginosa, for instance. So we ask whether this polymorphism occurs in UT-prone patients. And we investigate this in collaboration with the group of Katarina Svanborg in Sweden. And we found that first 5 of the 11 selected SNP were polymorphic in our population, and 2 of them, this one and this one, were associated with susceptibility to acute pyelonephritis. And it was really interesting to know that ancestral genotype for this polymorph, for this SNP, occurs in 30% in the pediatric control population for both, and are totally absent in UT-prone patients. It was also interesting to know that this paper showed that the same polymorphism were associated with susceptibility to Aspergillus fumigatus in patients undergoing bone marrow transplantation. And this haplotype, this polymorphism, were associated with increased mRNA stability and protein expression. So what this story showed is that after infection, PTK3 is rapidly expressed in a TLR4 mediated pathway, and then PTK3 opsonize UPEC, opsonize bacteria for very early elimination of UPEC by neutrophils and control of inflammation. Now I will move on cancer, and we will talk about the role of neutrophil in cancer. So the role of neutrophil in cancer today remains largely unknown. Why it's remained largely unknown? Because the role of neutrophil has been long viewed restricted to acute inflammation and restricted to elimination of pathogen and to phagocytosis. But today, this dogma has been challenged by several groups, and we know that neutrophils are present in tumor, and we know that neutrophils are involved in cancer-related inflammation, and it has been shown that neutrophils could display anti-tumor activity and pro-tumor activity. So anti-tumor activity mainly due to direct cytotoxicity of neutrophil toward tumor cells, mainly due to the production of reactive oxygen species. But one paper showed that TAN, so tumor-associated neutrophils, could stimulate T-cell response. In contrast, pro-tumor activity of neutrophil was related to angiogenic activity of neutrophil, notably through production of angiogenic factors such as VEGF. And PMN neutrophil can also cause DNA damage and genetic instability, also here mainly due to production of reactive oxygen species. And it has been shown that neutrophils, and here there is a link with the famous myeloid-derived suppressor cells, MDSC, you can have an inhibition of T-cell response. But today, the role of neutrophils, in vivo role of neutrophil in chemically induced cancer and inflammation-induced cancer remains unknown. So we decided to evaluate the role of neutrophil in tumorigenesis, taking advantage of GCSF receptor knockout mice. And GCSF is a growth factor for neutrophils, required for production of neutrophils, and also required for the release of neutrophils from bone marrow to circulation. So first, we confirm that these mice have a specific neutropenia. Other cells are not modified in terms of cell number in blood. And we analyze the susceptibility or the role of neutrophils in key model of inflammation-induced cancer and chemically induced cancer. So the first is 3-MCA-induced sarcoma, and the second is colitis-induced cancer induced by injection of mutagen AOM and AOM and then DSS in drinking water. So in the first model, we found that neutrophils deficiency, I will talk about neutrophil deficiency for GCSF knockout mice. Neutrophil deficiency was associated with increased susceptibility to 3-MCA-induced sarcoma, both in terms of tumor incidence, in terms of tumor volume. And this is bone marrow chimera, and bone marrow chimera reveals that expression of GCSF receptor in hematopoietic compartments was sufficient to rescue the phenotype. This is consistent with the fact that GCSF receptor is expressed on neutrophils. And it's important to note here that GCSF receptor is not expressed on cancer cells, and expression of GCSF receptor is very, very low on other leukocytes compared to neutrophils. So we are confident that the role of GCSF, that the phenotype is related to neutrophils. So then we decided to evaluate the inflammatory response, to compare the inflammatory response between wild type and knockout animals. First at early time points, so this is 10 days after 3-MCA injection. And this is inflammation found in the injection site. As you can see, inflammation was reduced in knockout mice. And we found reduction of TNF-alpha, reduction of IL-1 beta, also reduction of 6CL1, reduction of 6CL2. In contrast, other chemokine and cytokine were not affected, such as CCL2 and also interleukin-23. Then when we look at leukocyte infiltrates, this is infiltration of leukocyte in the injection site. So in the 3-MCA containing conor droplets. And as you can see here, recruitment of macrophages were slightly decreased. And we have an absence of neutrophil in the ejection site. So we next assess whether the phenotype was due to an excessive DNA damage, mainly due by possibly due to reactive oxygen species. So here by immunohistochemistry analysis, where we stand for 8-hydroxydeoxyguanosine, which it's a marker for DNA damage induced by reactive oxygen species, we didn't see differences between wild type and knockout mice. Here it's 10 days after injection. We also didn't see difference in expression, in production of reactive oxygen species between wild type and knockout animals. So phenotype is not due to DNA damage. We next study focus our activity on established tumor, and tumor, wild type and knockout tumor were harvested at the same volume, not at the same time, but at the same volume. And we assess for inflammation and polarization of immune response here. As you can see, total leukocyte number was similar in wild type and knockout mice, but microenvironment was different. Was different in terms of cytokine. We found decreased expression of interferon gamma. Interferon gamma is well known for its anti-tumor activity. We also found decreased expression of IL-23, IL-1 beta. This molecule are expressed by myeloid cells and are also known to be expressed by neutrophils. We don't know if it's due to lack of expression of neutrophil, but we also found strong increased expression of 6CL1, GCSF, of course, because you don't have a receptor, and SDF1. It's interesting to note that these chemokines are important for recruitment of neutrophils. And neutrophil express the receptor, of course, so maybe the lack of receptor is due to this increase of expression. Other cytokine and chemokine are not modified. And then we have a look on the subpopulation of leukocytes infiltrating the tumor. Here it's neutrophils, so 10% of leukocytes in wild-type mice in tumor-infiltrating leukocytes are neutrophils, but they are absent in knockout animals. In contrast, macrophages were increased, and other cell population were not affected. And of course, we decided to investigate the polarization of this neutrophil, of this macrophages. Sorry, and this is strategy to get macrophages in tumor. And this cell population were sorted by FACS, and we analyzed expression of selective marker by real-time PCR, so M2-like marker and M1-like marker. Just remember that M2 link is related to angiogenic activity, protumoral activity, and M1 is related to anti-tumor activity. And as you can see here, the balance between M1 and M2 is clearly skewed toward M2-like polarization in neutrophil deficient mice. Almost all molecule, all M2-like M2 marker were increased in knockout in neutrophil deficient mice, whereas M1 marker was unaffected or decreased in tumor-associated macrophages isolated from neutrophil deficient mice. Then we decided to study the polarization and maybe activation and maybe polarization of tumor-associated neutrophil. Of course, only on wild-type animals, because you don't have neutrophils in knockout mice. And first, we decided to assess whether recruitment of neutrophils into tumor bed could be associated with an anti-tumor response. And we assess whether an association exists between density of neutrophils and proliferation state of tumor cells. And as you can see here, we found that higher density of neutrophil was associated with the area of low proliferative states in the tumor, suggesting that recruitment of neutrophil is associated with anti-tumor immunity. Then here by FACS analysis, we analyze the activation state of neutrophils by two markers. The first is CD62 ligand, so L-selectin, and decrease of L-selectin expression of neutrophil is an hallmark of neutrophil activation. And in contrast, increase of ICAM1, CD54, is relatively new, but it has been correlated with anti-tumor activity of neutrophil and one polarization. So as you can see here, we found that tumor-associated neutrophil compared to bone marrow neutrophils acquired this phenotype. Then tumor-associated neutrophil were sorted by FACS, and we analyzed expression of selective marker. We compare expression to naive neutrophil, and as you can see here, we found increased expression of interesting marker of interferon gamma, MET, NOS2, and CCL3. This marker has been related to N1 polarization and anti-tumor activity in neutrophils. In contrast, CCL5, which has been associated with N2 polarization and pro-tumor activity of neutrophil, wasn't changed. So this showed that neutrophils had activated phenotype, and has been is associated with anti-tumor activity. So we ask whether neutrophil has direct anti-tumor activity. And this is a response: weekly adoptive transfer of bone marrow naive neutrophils in knockout mice when the tumor became palpable, so here, delayed, reduced, or totally delayed the tumor growth. And what you found also, it's a rescue of interferon gamma production in mice. So this clearly showed that neutrophils was still with anti-tumor immunity, and neutrophil has direct anti-tumor immunity. What about the second model? So model of RMDSS-induced colorectal cancer. Also here we found that neutrophil deficiency was associated with increased susceptibility in terms of polyp number, here, in terms of body weight loss, in terms of colon length shortening, and in terms of bleeding score. But this model here has a peculiarity, and the peculiarity has been described by several groups, in particular by the group of Richard Flavel, that intestinal dysbiosis, so intestinal microflora, play an important role in this model. And intestinal dysbiosis has been associated with colorectal cancer. When we have a look in the literature about associations of the role of neutrophil in to control microbiome, it was unknown. So we tested this hypothesis, and what we found is that here, as you can see, in fact, that neutrophil deficiency was associated with dysbiosis. This is beta diversity in genus level, so in knockout and wild-type animals. And consistent with this data, experiment of co-housing experiment here showed that you totally lost the phenotype in terms of body weight loss and in terms of polyp number. So this strongly suggests that neutrophils control intestinal microflora, and this is due to this induce this phenotype. This is the conclusion of this study. So neutrophil deficient animals showed increased susceptibility to 3-MCA-induced sarcoma. We found that tumor-bearing neutrophil deficient mice showed altered tumor microenvironment, polarization of tumor-associated macrophages in M2-like phenotype. Neutrophils in tumor had activated phenotype, and higher density of neutrophil was associated with area of low proliferative activity of the tumor. And adoptive transfer of neutrophils after tumor onset had anti-tumor activity. In colitis, we found that neutrophil deficient mice showed dysbiosis and increased susceptibility to our MDSS-induced cancer. And we found that protection from colitis and colitis-related cancer in neutrophil deficient mice can be achieved by co-housing experiment. Now, if I have 5 or 10 minutes more, I will talk about human cancer. So this is a new story that we published. I think 2 months ago it was published on PubMed, but it's published this month on the journal. What is the occurrence and clinical significance of neutrophil in cancer? In general, clinical significance of neutrophil in human cancer has been associated with poor prognosis. And this is the data. But what about colorectal cancer here? As you can see, clinical significance of neutrophil in colorectal cancer, in human colorectal cancer, has been object of conflicting reports. And this is probably due to different methodological approaches. So our aim was to define a reliable methodological approach, quantify TAN in colorectal cancer tissue, and to assess clinical significance of neutrophil infiltration in colorectal cancer. This study has been done in collaboration with the group of Luigi Laghi here. So first, we compare two markers, so myeloperoxidase and CD66B, which has been used in the literature for years and years to stain neutrophils. But if you stain, double staining in colorectal cancer tissue for MAPO and CD66B, what you found here is that all CD66B positive cells were MAPO positive. So these cells are clearly neutrophils, no problem. But what we found also, it's MAPO positive cells, CD66B negative cells. And here, if you stain MAPO and CD68, which is a marker for macrophages, you found a double positivity. So it means that these cells are probably monocyte hematomacrophages. So clearly, this shows that MAPO cannot be used to stain neutrophil in tissue, at least in colorectal cancer tissue, but I'm sure that it's a general rule. So we decided to evaluate density of TAN by using CD66B. So this is our case list. It was patient who underwent surgery from colorectal cancer here in Humanitas. And density of neutrophil was blindly assessed by immunostaining, here followed by computer-assisted measurement of immunoreactive area. So this is data that you obtain. So median value of neutrophil immunoreactive area were used to separate population between low and high. And data were entered in the Cox proportional Hazard model to evaluate potential impact of outcome. And events that we follow was death of the patient for DSS, disease-specific survival, or relapse of cancer for DFS, for disease-free specificity. So as you can see here on this model, high TNM stage, TNM is for tumor non-metastasis. It's a classical staging system used in clinic, and it's based on anatomical extent of the tumor. So this TNM stage and vascular invasion were significantly associated with worse outcome in cancer, as expected, of course. But what we found, it's also high neutrophil density, both in the invasive margin and intratumoral compartment, was associated with better clinical outcome and better DFS and better DSS. This is hazard ratio, so 0.5, 0.3, 0.4, 0.5. So the probability that the events occur. This is a Kaplan-Meier curve that we obtain. As you can see, it's clear that you found a significant better outcome in patients with high infiltration of neutrophil in red compared to low infiltration of neutrophil in black. Then we don't stop our analysis here, and we ask whether this potential prognostic value could be influenced by other variables. So we enter data in a multivariate analysis, and what we found here is that we lost the significativity for neutrophil. Okay, only pathological stage, so TNM stage, and vascular invasion were independently associated with clinical outcome. But what we observe here, that there is a very nice interaction between 5-FU treatment and TAN density. So we decided to restrict our analysis on our population on stage 3 colorectal cancer patients, which are patients for whom it's expected to gain the highest clinical benefit from 5-FU therapy. So here we have 178 patients, and as you can see here, infiltration of neutrophil and density of neutrophil per se was not associated with a better outcome, even if we have clear tendency. But also here you found a high interaction between 5-FU and neutrophil density. And this is what we obtain if you separated patient between treated patient, non-treated patient, and all patient. So clearly you found that high density of neutrophil was associated with better clinical outcome in treated patient, but not in non-treated patient. And this is just the mean of the both population. And this data claim that dual clinical significance of neutrophils was observed in colorectal cancer patients. So now let me thank people involved in this study. First of all, Professor Alberto Mantovani, of course, also Cecilia Galanda, our PI, all the group that participated to the study, and in particular Andrea Ponceta for the story of mouse cancer, Maria Rosaria Galdiero also for the story of clinical outcome, and all the group of Luigi Laghi and Fabio Glizzi also for the story of clinical outcome, and all our partners. Thank you very much also for your attention.